Okay, so in this video, we're going to talk about the small intestine, the large intestine, and chemical digestion. So first, we're going to talk about the small intestine. The small intestine is the area where most of the chemical digestion and absorption of nutrients occurs. About 90% of the nutrients are going to be absorbed in the small intestine. They're absorbed from the small intestine and into the bloodstream in the body. The remaining 10% will be absorbed in the large intestine. Another name for the small intestine is the small bowel. The small intestine is 15 to 25 feet long. It has a diameter of about 1.5 inches up by the stomach, and that narrows down to about 1 inch by the time it gets to the large intestine. The small intestine can be found in all the abdominal regions except for the hypochondriac and the epigastric regions. The small intestine is stabilized by that mesentery that we named the mesentery proper. The small intestine is supplied by an artery called the superior mesenteric artery, and it is drained by a vein called the superior mesenteric vein. And remember, the superior mesenteric vein joins with the inferior mesenteric vein to become the hepatic portal vein. There are 3 segments of the small intestine: the duodenum, the jejunum, and the ileum. The duodenum is the first section of the small intestine, and it's about 12 inches long. It receives secretions from the pancreas and the liver, and it also receives the chyme from the stomach. The duodenum is in the shape of a C, and right where it curves, that is where the pancreas sits. The duodenum is considered retroperitoneal because it's mostly behind the peritoneum. The duodenum is often called the mixing bowl because all of those secretions from the stomach and the pancreas and the liver end up in the duodenum and they just get mixed in there. But because the duodenum is so short, there's not a lot of chemical digestion that will actually occur there, and there's not a lot of absorption either. The contents in the duodenum then move into the next section of the small intestine, which is called the jejunum. The jejunum is about 8 feet long, and this is where the bulk of chemical digestion and absorption occur because it is so long and because there are all of these prominent circular folds. The circular folds add surface area and allow more absorption to occur there. The third part of the small intestine is called the ileum, and the ileum is about 11.5 inches in length. It ends at what is called the ileocecal valve, and this is a circular sphincter muscle between the ileum and the cecum, which is the first part of the large intestine. The ileocecal valve controls the flow into the cecum. Let's talk a little bit about the histology of the small intestine. So the small intestine has these circular folds. These folds are permanent folds. They're unlike the rugae of the stomach. The rugae in the stomach could flatten out when the stomach became distended. The circular folds help to increase surface area for absorption, and each fold has finger-like projections on it called intestinal villi. Here's a picture of one singular villus. We call these villi the intestinal villi. The intestinal villi are lined by simple columnar epithelium. These simple columnar cells have microvilli on them that look like the bristles of a brush. So we call this the brush border. The combination of the brush border, the intestinal villi, and the circular folds increase the surface area to 2,200 square feet. That's about 10 lanes of a bowling alley. Underneath the epithelium is the lamina propria. The lamina propria contains capillaries that are going to absorb the nutrients. Each villus also contains a lacteal. The lacteal is a lymphatic vessel. It's the end of a lymphatic vessel, and the lacteal will be used to transport materials that can't enter the blood. For example, amino acids, anytime you eat proteins that get broken down into amino acids, they will be absorbed into those simple columnar cells, and then they'll be absorbed into those capillaries. Same thing with glucose. They get absorbed from the gut into those simple columnar cells and then into those capillaries. However, fatty acids are too large for the capillary, so they get absorbed into those simple columnar cells, and then they have to be absorbed into the lacteal. The lacteal merges with other lymphatic vessels to become the thoracic duct. The thoracic duct then drains into the left subclavian vein. So all of those fatty acids are bypassing the liver and they're going directly into the blood. The capillaries, however, that contain the amino acids and the glucose, they are going to merge into the hepatic portal vein, which will go to the liver, and then the liver can decide how to metabolically regulate those. Do they need to be stored, or can they be put out into the general circulation? At the base of the villi are intestinal glands. These intestinal glands are also lined by these epithelial cells, but in between these epithelial cells are other cells. There are stem cells that produce new epithelial cells when they get shed, and when those epithelial cells get shed into the lumen, they will release their enzymes into the rest of the contents of the intestine. There's also Paneth cells. Paneth cells release defensins and lysozymes, and they are part of the innate immunity and will help to protect the body from any foreign pathogens in the intestine. Then there are these enteroendocrine cells that are also in between those epithelial cells. Those enteroendocrine cells are going to secrete hormones. And we've talked about these hormones already, but these hormones are gastrin, CCK, and secretin. Beneath the mucosa is the submucosa. The submucosa contains duodenal submucosal cells that secrete mucus. They secrete copious quantities of mucus, and the mucus will protect the lining from acids. It also contains bicarbonate, so it'll raise the pH of the contents inside the intestines. Each segment of the small intestine has its own specialization. In the duodenum, there are fewer circular folds, so there will be little to no absorption. There's also smaller villi. The main function in the duodenum is just to receive the chyme and to receive the secretions from the pancreas, gallbladder, and liver. Its main job is to mix all these things together and to neutralize the acids in the chyme before it reaches the jejunum. The jejunum has very prominent circular folds and villi. More so in the first half than in the second half. They start to decrease in size the closer they get to the ileum. So most of the absorption is going to occur in the first half. One drastic weight loss method is to remove the first half of the jejunum so that there's less absorption of fats and calories. Most chemical digestion and absorption takes place in the jejunum. At the distal portion of the ileum, there are no circular folds, so no absorption will be occurring at the distal end of the ileum. But there are these lymphoid tissues called Peyer's patches, and they help to protect the small intestine from bacteria from the large intestine if it should back up into the small intestine. The cells in the glands of the small intestine produce 1.8 liters of intestinal juice every day. The fluid in the intestinal juice comes from the mucosa through osmosis, and it also comes from the glands through secretion. The intestinal juice helps to moisten the chyme, and it helps to buffer acids, and it just keeps all those nutrients and enzymes in solution. In addition to the mucosa and the glands, there are those brush border cells, and those brush border cells release enzymes that break down material that comes in contact with it, so then the epithelial cells can absorb it. So an example of the secretions of a brush border cell is enteropeptidase. That's a brush border enzyme. Enteropeptidase will activate the proenzyme called trypsinogen. There's also submucosal glands in the duodenum that begin secreting during the cephalic phase through the vagus nerve. The mucus helps to protect the duodenum from upcoming assault of acids. The sympathetic nervous system inhibits the duodenal cells, which means there will be less mucus being produced, and that leaves the duodenum unprepared for acids. So in cases where there's a person that has chronic stress, they're not producing enough mucus, they're not protecting their duodenum, and that could lead to ulcers. It takes about 5 hours for food to move through the small intestine from the duodenum to the ileum. So when you eat breakfast, the food's going to leave the small intestine by about lunchtime. The movement of the mucosa enhances the absorption, so the movements help to stir and mix the contents and make the absorption more efficient. So now we're going to move into the large intestine. Another name for the large intestine is the large bowel. The large intestine has 3 segments to it. It has the cecum, which is the first segment, the colon, which is the second segment, and the rectum, which is the last segment. The cecum is an expanded pouch. The ileocecal valve controls what enters into the cecum, and then there is the appendix, which is attached to the cecum. The cecum stores materials coming in from the ileum, and it begins compaction of those materials. The appendix contains lymphoid nodules. The colon is thinner and wider than the small intestine, and it contains these pouches that are called haustra. The haustra permit the colon to expand. The haustra are produced or created by these smooth muscles along the length of the colon called tinea coli. The tinea coli muscles have a muscle tone to them, which creates the haustra. The colon is divided into 4 segments: the ascending colon, the transverse colon, the descending colon, and the sigmoid colon. The ascending colon starts at the cecum, and it travels up to just underneath the inferior part of the liver, then it bends. Where it bends, we call it the right colic flexure. The transverse colon extends from the right colic flexure across the abdominal cavity to the left colic flexure. It's horizontal, and at the left colic flexure, it bends to become the descending colon. The descending colon descends down to the iliac fossa, and then it curves towards the midline to become the sigmoid colon. The sigmoid colon then empties into the rectum. The entire large intestine receives blood from the superior and inferior mesenteric arteries, and then they are drained through the superior and inferior mesenteric veins. Colorectal cancer is cancer of the colon and or the rectum. It is the 3rd most common cancer, And the best defense against this cancer is early detection and then prompt treatment. We have screens that will screen the feces for blood, and then colonoscopies are also used to detect colorectal cancer. The rectum is 6 inches long and it stores feces. The anus is at the end of the rectum where the feces exit. So let's take a look at the rectum. The rectum has these internal anal sphincter muscles. They are smooth muscles and they're involuntary. So they're going to relax whenever there's pressure inside the rectum from the presence of feces. Then we have the external anal sphincter muscles, and those are skeletal muscles that guard the anus, and they're going to stay closed until you voluntarily open them. In the lamina propria and the submucosal layers of the rectum, there are a network of veins, and when those veins become distended, that's called a hemorrhoid. So the large intestine has no villi, but it has many goblet cells. The goblet cells are in these deep intestinal glands that are deeper than what are in the small intestine. And these intestinal glands are just dominated by these goblet cells. The goblet cells produce a mucus, and the mucus provides lubrication to help the dry fecal material move through the intestines. These goblet cells are stimulated to begin secretion whenever there's any type of local stimulation. The friction and the harsh chemicals of the materials that are coming into the large intestine are going to stimulate those goblet cells to start secreting mucus. Any digestion that's occurring in the large intestine occurs because of bacteria. Less than 10% of the nutrient absorption, though, is going to occur in the large intestine. The other 90%, like we said, occurs in the small intestine. So the colon is mixing and propelling food just like the small intestine, and it prepares the fecal matter for defecation. Like I said before, absorption of water and nutrients and toxins, most of it occurs in the small intestine, like 90%. But the large intestine also absorbs water. Of the 1,500 milliliters per day of material that enters the large intestine, only 200 milliliters will be ejected as feces. So quite a bit of it is still being absorbed. The feces will be 75% water, 5% bacteria, and the rest is indigestible material, inorganic matter, and epithelial cells that are sloughing off from the large intestine. Other things absorbed from the large intestine back into the body are things like bile salts, vitamins, organic wastes, toxins. There are bacteria that are normally found in the large intestine, and so these are friendly bacteria, and they generate 3 vitamins in the large intestine. They generate vitamin K, biotin, and vitamin B5. Vitamin K is a fat-soluble vitamin that's required for producing clotting factors, including prothrombin. Biotin is needed for glucose metabolism, and vitamin B5, otherwise known as pantothenic acid, is needed to make steroid hormones and some neurotransmitters. A vitamin K deficiency can lead to impaired blood clotting. A vitamin K deficiency can occur from a diet that's lacking dietary fats, or if there's inadequate bile, or if a person is having diarrhea and the vitamin K isn't being absorbed into the body. Deficiency of biotin and vitamin B5 are more rare. Through bacterial action, bilirubin, which is a waste product that the liver secretes into the intestines, is converted to urobilinogens and stercobilinogens. Stercobilinogen becomes stercobilin in the colon, and that is what causes the feces to have a yellow-brown color. Stercobilinogen is converted to stercobilins when it's exposed to oxygen. Bacterial action also breaks down peptides in the feces, and this generates ammonia. We talked about this before. The ammonia and other products of the breakdown contain nitrogen, and nitrogen gives off the odor of feces. It also contains hydrogen sulfide, and that gives that gas with a rotten egg odor. Most of the ammonia is reabsorbed back into the hepatic portal system, where the liver then converts them to that non-toxic substances like urea so that the kidney can excrete them. Indigestible carbs enter into the large intestine intact, and the large intestine really has no enzymes to break them down, so the bacteria use them as a nutrient source, and as they're metabolizing them, they create gas, which is called flatus, in the process. Foods with large amounts of indigestible carbs, like beans, stimulate gas production, and sometimes gas production can lead to distension or cramps or a discharge of gas. Now let's talk about motility and the defecation reflex. Movement from the cecum to the transverse colon is pretty slow. It takes some time to absorb the water, and then the material gets converted to sort of a sludgy paste. The haustra start churning and they mix the contents. The large intestine receives a message from the stomach and the duodenum, which signals the nerve plexus in the large intestine to cause these mass movements, which are powerful peristaltic contractions. With these powerful peristaltic contractions, feces move into the rectum and initiate what we call the defecation reflex. These powerful peristaltic contractions force the feces into the rectum. The rectal chamber is usually empty. When mass movements forces the feces into the rectum, an involuntary defecation reflex results in bowel movement. The defecation reflex is involuntary. So it's involving the internal anal sphincters. I now just want to talk briefly about chemical digestion. So we're going to talk about the individual chemicals. So when nutrients are brought into the GI tract, they have to be broken down into the smallest units possible in order to be absorbed into the body. So when they're brought in, they are polymers, and they have to be broken down into the smallest unit, which we call monomers. So, for example, a carb is a polymer, and it has to be broken down into a simple sugar, like glucose. And a protein is broken down into its monomer, which is amino acids. And lipids are going to be broken down into their monomers, which are fatty acids. We also have nucleic acids, but they're not a big part of the nutrient base that we have. But nucleic acids get broken down into nucleotides. It's digestive enzymes that start to break down those polymers into those monomers. And then after that, the brush border enzymes will break them down even smaller and will break down whatever remains. So we're going to look at each of the individual nutrients. We're going to start by looking at carbohydrates. So carbohydrate digestion begins in the mouth. That's from salivary amylase. It starts breaking them down. And remember, that's from the parotid and the submandibular glands. And it breaks the complex carbs into di and trisaccharides. So disaccharides and trisaccharides. And it continues until they get down to the stomach. And then once in the stomach, they stop because the pH is too low. So amylase stops working. And there's nothing that's going to happen in the stomach, but then once the food gets down to the small intestine, so into the duodenum, then we have the pancreas dumping alpha amylase into the duodenum. So then the pancreatic alpha amylase breaks down the carbohydrates. It's the same exact amylase. So we had salivary amylase in the mouth, and now we've got pancreatic amylase. It's the same amylase. It's just that it's coming from a different location. In the mouth, it's coming from the salivary glands. In the duodenum, it's coming from the pancreas. So the amylase starts to break these carbs down into, again, those di and trisaccharides. Those di and trisaccharides will then be broken down into monosaccharides. And they are fragmented into monosaccharides by these brush border enzymes. The brush border enzymes are from the cells at the base of the intestinal microvilli. There's a couple of different types of brush border enzymes. There's maltase. That will break the bonds between two glucose molecules that make up maltose. There's also sucrase. And there's also lactase. So maltase breaks down into two glucose molecules. Sucrase breaks down into glucose and fructose. Those are both monosaccharides. And lactose breaks down into glucose and galactose. right? So all of these things, these are all monosaccharides, and they can all be absorbed into the body. So these nutrients then will get absorbed into the body. And they're absorbed into the body using a facilitated transport. So facilitated transport just means that it's going to use a protein to carry it across the cells of the intestinal wall. And then once it crosses the cells of the intestinal wall, it's then going to, those nutrients are then going to move into the blood. So they will get transported into the capillaries. And the capillaries then will carry those nutrients off to the liver. And the liver will then decide what to do with them, whether to store them or whether to send them out into the general circulation so the other tissues of the body can use them. Let's look at lipid digestion. So there is some lipid digestion in the mouth. If you remember, there was lingual lipase. So that starts to break down some of the lipids, but again, at the stomach, because of the pH, the lingual lipase stops. Once in the duodenum, though, there are a couple of things that are going to help to break down those lipids. First of all, we have the gallbladder, which releases bile. And bile, if you remember, it emulsifies fats. So it's going to take these large lipids that need to be broken down into individual lipids, into tiny droplets. And then those tiny droplets then can get broken down by pancreatic lipase. So pancreatic lipase breaks down these droplets into triglycerides. Remember what a triglyceride looks like. It has a glycerol head and 3 fatty acid tails. And then the bile salts are going to interact with these triglycerides. So you have bile salts coming back into play, and they form these little micelles with the triglycerides. The micelles are arranged where the heads are facing outward because they're hydrophilic, and the tails are facing inward because they're hydrophobic. So this is what a mycele looks like. And then the mycele then gets absorbed into the intestinal cell. So these are the cells lining the intestinal wall, and they get absorbed into those cells. And once in the cell, then they get broken down again into the triglycerides. So it's just a way for them to get absorbed as the mycele, and then they're broken down into the triglycerides again. Once in the intestinal cell, those triglycerides get coated by a protein. So a protein is going to surround that triglyceride. It gets a protein coat, if you will. And now we're going to call this a chylomicron. The chylomicrons then are going to be absorbed into the body, but instead of going into the blood, they're going to go into the lymphatics. So there's a lymphatic capillary, and that's where those chylomicrons are going to go. And once they're in the lymphatic system, then they will head back to the heart and be dumped into the subclavian vein. So eventually then, because the subclavian vein leads into the heart, and the heart pumps that blood back out again, that subclavian vein will end up bringing those lipids, those chylomicrons, to the liver. and then the liver has a purpose for those chylomicrons. So this is just a little bit more complicated way of how those lipids are absorbed into the body. Now we're going to talk about protein digestion. So protein digestion is complex and it's very time consuming. There's mechanical processing that occurs in the mouth and in the stomach and the intestine, just like with the other nutrients. And then there's also chemical processing as well. But it's really, protein digestion doesn't really start until the stomach. So even though the mouth is churning and breaking it down, we're still not getting down to the monomers, to the smallest unit that can be absorbed. The pH of the stomach is low. It's like 1.5 to 2.0. So that's very, very acidic. And that's because there's a lot of hydrochloric acid that's being secreted in the stomach. Well, the hydrochloric acid allows the conversion of pepsinogen to pepsin. Okay, so remember, pepsinogen is secreted by the chief cells. And pepsin then functions very well at the pH of 1.5 to 2.0. Pepsin breaks down proteins, otherwise known as polypeptides, into smaller peptides. So pepsin is from the stomach. And now these smaller peptides are going to move into the duodenum. And in the duodenum, the pancreas secretes enzymes into the duodenum. One of those is called the pancreatic protease. So these pancreatic proteases are in an inactive form and they have to be activated. So once they become activated and they're active, then they can break those smaller dipeptides down into dipeptides. So we're just getting smaller and smaller. Now a dipeptide is just two amino acids linked together. Now these amino acids, these dipeptides, come in contact with the epithelial surface. And on the epithelial surface, there are these enzymes called brush border enzymes. And they will break down those amino acid, those dipeptides, into individual amino acids. So it's quite a process, right? So the stomach starts out by breaking down proteins into polypeptides and then into smaller polypeptides. And then the pancreatic proteases break down those polypeptides, those smaller polypeptides, into dipeptides. Then we have the brush border enzymes that are on the epithelial surface that line the small intestine, and they're going to break down those dipeptides into individual amino acids. Now those amino acids then can get absorbed into the bloodstream. So they're going to go into the bloodstream, and now we have amino acids in here, and those amino acids are going to go straight to the liver, and then the liver will decide, do we want to store those amino acids? Do we want to make proteins out of those amino acids and store them? Or do we want to send amino acids into the blood? And if we send amino acids into the blood, now all the tissues in the body can enjoy those amino acids. Again, those amino acids from the blood, from the small intestine, are going to get delivered to the liver through the hepatic portal vein. If you remember what the hepatic portal vein is, it's the emergence, so that's the mergence of two other veins called the superior mesenteric vein and the inferior mesenteric vein. So now just briefly, I want to talk about water absorption. So you have about 2 to 2.5 liters of water that enters into the digestive tract through the food that you eat and the water that you drink or the whatever drinks that you drink. And then we also have some water that's produced by your glands in your body. So salivary glands, gastric glands, intestinal glands, other glands, they produce about 6 to 7 liters of water a day. So those are all from the glands in your body. The 2 to 2.5, that's from what you eat and drink. So total, you have, anywhere from 8 to 9.5 liters of water that's being absorbed every day. This water absorption, of course, is through osmosis. And the reason it occurs is because first sodium and chloride, or salt, gets reabsorbed, and then the water just follows it. So that's why if you have a really salty meal, water's going to follow it and you end up with swelling. The other thing that gets absorbed are vitamins. There are 9 water-soluble vitamins. All of them are easily absorbed by the digestive epithelium and then into your body. The only one that's difficult to absorb, or it takes a little extra work, is vitamin B12. And if you remember from the video that you watched, vitamin B12 needs to be bound intrinsic factor in order to be absorbed into the body. And if you remember, intrinsic factor is secreted by parietal cells in your stomach. Without the intrinsic factor, you're not going to be able to absorb vitamin B12. So it doesn't make any sense for patients who lack intrinsic factor to take vitamin B12 by mouth because it's not going to get absorbed anyway. Those people would have to have vitamin B12 injected directly into their blood. That ends this chapter then for the digestive system. So I will see you next time.